Last week was a rough. I mean, after like almost a month of not being in person, it was rough.
Yeah, I used to rush you.
Yeah.
All right. So that wasn't a whole ton of time. We'll just kind of start working on this together. So at the end of interphase, our chromosomes have replicated. So instead of looking like single chromosomes, they should look something like this, right? And when they replicate, they're an exact copy of each other. So this chromosome that's replicated will have both of the dominant copies, right? And this chromosome that's replicated should have both of the recessive copies. So during interphase, our DNA is copied, right? So now each chromosome is replicated, has two identical sister chromosomes. Metaphase 1, what happens in metaphase 1? In any metaphase, what happens? The chromosomes line up in the middle. The special thing in meiosis is that when they line up the first time, they're lining up in pairs, in tetrads, right? So the homologous chromosomes have paired up. I only have one set of chromosomes here, so. So one will be on this side, the other one will be on the other side of the plate, right? Metaphase is a really important stage because there's a checkpoint that happens here in metaphase, right? Because this is what decides what goes to what cell. So the chromosome that lines up on this side of the plate is going to go that way, and the chromosome that lines up on this side of the plate is going to go that way, right? We also want to show that crossing overs happen. So what happens during crossing over? Yeah, sister chromatids can swap alleles between the homologous chromosomes. So which do you want to swap, the H or the A? The A. So if we swap those A's, right, then in metaphase, one chromatid will have both dominant copies, and this chromatid will have the dominant H and the recessive A, and this one will have the recessive H and the dominant A. I'm sorry, this is really small, so you probably can't see that. And then the fourth one will have both recessive copies, right? So it'll be HA, dominant, recessive, dominant. So those chromosomes are eventually going to end up in two separate cells at the end of meiosis 1. So the next step that really determines what's going to go in each new cell is again at the end of metaphase 2. So again, in metaphase, what happens always in metaphase? They line up in the middle. At this point, we only have one chromosome in each cell, so one sister chromatid is going to be on one side, one sister chromatid is going to be on the other side. So this chromatid is going to end up in a cell. This chromatid is going to end up in a cell. This chromatid is going to end up in a cell. That chromatid is going to end up in a cell. One of the other things you have to be able to fill in is this notation box. So in this case, we're starting with a cell that's 2n equals 2. The 2n means that we have two copies of each, that it's diploid. So we have two homologous chromosomes here. Okay. At this point, the notation should be still 2n equals 2. At this point, we're still 2n equals 2. It's not until after meiosis 1 is done that we're down to being haploid. Yeah, 1n equals 1. And each of these new cells is also going to be 1n equals 1, right? So it's haploid, and we have half the number of chromosomes that we started with. So we start with a diploid cell. We end up with a haploid cell. Each one of these gametes is genetically unique, right? They each have a different combination of alleles. That's the whole point of meiosis, that we end up with a haploid cell with a unique genetic composition. Questions on that? You will have to pretty much do that exact same drawing on the quiz on Thursday. So you need to know why it's drawn that way. You need to be able to replicate that with similar alleles. Okay. Questions? All right. So we are going to move on a little bit in some notes today, and then I'm going to give you the rest of time to finish up your lab from Friday. So if you haven't pulled out notes yet, you can do that. Hopefully you wrote that down somewhere. Last topic, last section of this particular unit on cell division has to do with chromosome abnormalities. And these are errors that occur somewhere in the cell cycle. So either in interphase or in the division process itself. We're specifically dealing with meiosis here. So these are changes that are going to get passed on in our gametes. So that's the list we're going to go through. And just to kind of warm up for discussion, each of those blue things there, go ahead and take some time talking about what you think might be happening during those things. So they're named for what happens. What's going to happen. We could probably briefly touch on this in Bio 1, but I know it's been a while. We're going to look at those changes in chromosome structure first. And so these are happening like within a chromosome. So generally when the chromosome is being copied, or sometimes this can happen during crossing over. Okay, so the first two are errors that occur during replication. So the first one of those that happens during replication is deletion. So it's exactly what it sounds like. We're going to lose a chunk of our chromosome. So when we copy it, we miss copying a section of it. So the new chromosome's missing a piece or part of it. And so each one of these is going to be accompanied with a diagram. Take a couple of seconds to talk about in your group, why does this diagram show deletion? What's going on there? So in this case, this is very, so we have way more genes than this on every chromosome, right? But we have these genes A, B, C, D, E, F, G, H. In this case, when we copied it, we missed copying this section. And so now that this chromosome does not contain that, the copy of that chromosome does not contain the D gene. Okay? So it's a deletion when we lose a part of a chromosome, and we can lose a gene or multiple genes in that process. Okay, so that's deletion. We can also copy things on accident twice. So in duplication, when we copy the chromosome, sometimes it gets reset. We have to only make 2 copies of the gene. That's duplication. So again, there's a diagram there, just really quickly with your group members. How is this showing a duplication happening? So result-wise, if this happens, and you kind of have to keep in mind that we're actually getting 1 copy from each parent, right? But the copy we get from this parent is going to be missing a gene, which means we're not going to be able to make that protein. Okay, so we might have 1 working copy from the other parent, but this parent is not going to pass on a working copy of the gene if they get that chromosome. So we're not able to make that protein D from that information. Right? What could potentially happen as a result of this? Remember, these genes are going to get used to make protein, so we're going to end up making potentially twice as much protein as we should be. Okay? And so that can cause problems too. So not making a protein obviously can cause problems. Making too much of a protein can also cause problems. It's going to jam things up. So that's deletion and duplication. Those are mistakes that happen when we copy the DNA. There are also, because we cut and reconnect DNA during crossing over, there's a couple of things that can, incidents that can happen here. Okay, so the first one of those is inversion. So the word inversion just means to flip, right? So inversion, when we cross over, in trade pieces of chromosome, sometimes the piece of the chromosome gets flipped. So again, there's a diagram there. Take a couple seconds, talk about that with group member. How can you see inversion happening? So instead of going A, B, C, D, E, F, G, H along the chromosome, it now goes A, B, C, B. So this darker purple section here, the genes got flipped around. Okay. Now, depending on where we make the cuts, this might not be an issue. You can make the proteins no matter what strand of the DNA they're on. So if we cut in the right place, so if we cut between introns, it's not going to be an issue potentially. But if you cut right in the middle of the gene and you flip-flop it, then you could end up having genes on either end of the inversion that aren't working properly, that aren't creating proper protein. And the last one of these is translocation. So sorry, it's getting kind of low down to the screen, but translocation, we're moving a section of the chromosome to a different non-homologous chromosome. Moving a segment from one chromosome to another, again, specifically from one chromosome to another chromosome that is not homologous. We're putting genes on a different chromosome. From a chromosome 2 to a chromosome 21, or from a chromosome 21 to a chromosome whatever number. So this isn't the best example, because really this should be showing starting with 2 different chromosomes and ending with 2 different chromosomes, because we're moving one part to a different chromosome. But you can see here this AB gene got switched with an MNO gene that would originally have been on a different chromosome. The AB is going to end up on the other one. Translocation, we're moving genes around from one chromosome to the other. Okay, again, you're still going to be able to make all the same genes if it gets cut in the right place. However, when those genes get separated and moved out to other cells, it might mean that one cell gets 2 copies of that particular section, and the other cell gets no copies from the original that original gene. So we'll kind of take a look at that more in a little bit. So questions on any of those changes in chromosome structure? I think the names are pretty straightforward, but it's also important to think about what their effects are going to be. Are we not able to make a protein? Are we making too much of a protein? So the other big issue that can happen in the process of meiosis is something called non-disjunction. So take a look at that diagram. And non-disjunction can happen in both stages. It can happen in meiosis 1, and it can happen in meiosis 2. So with your group members really quickly, what do you think non-disjunction is? What's happening there? Non-disjunction. Yes, what do you think disjunction means? Separation. Disjunction means to separate. So non-disjunction means that they do not, in fact, separate. Okay, so it's kind of a double negative there. They don't separate when they're supposed to. So in meiosis 1, the homologous chromosomes are supposed to separate. In non-disjunction, they don't. Okay. In meiosis 2, the sister chromatids are supposed to separate. If non-disjunction happens, they do not. So non-disjunction occurs when chromosomes don't separate properly in anaphase. So normally, if we were doing this process and non-disjunction wasn't an issue, we're starting out with a cell that's diploid and has 4 chromosomes. Ideally, what should our final gametes end up being? What should the notation be normally, under normal conditions? 1n equals 2. Okay. We should end up with 1 of each of these chromosomes in our cell, right? In this case, 1 short, 1 long. If non-disjunction happens, though, if it happens in meiosis 1, both of these gametes are going to end up being 1n equals, well, these cells get an extra copy, and these cells end up only having 1 copy, right? So we have 1 too many chromosomes here. We have 1 too few chromosomes here. So these cells have 1 too many. These cells have 1 too few. If non-disjunction happens later on in meiosis, half the gametes are still going to be okay because they went through meiosis 1 correctly. The other half of the gametes, one's going to have 1 too many, one's going to have 1 too few. Okay, because remember, we talked about how my metaphase is an important stage. Okay, so anything on this side is going to end up in a different cell. So, with the group members, compare and contrast the results if the non-disjunction happens in meiosis 1 versus if the non-disjunction happens in meiosis 2. Yeah, so I would just say if it happens in meiosis 1, then all of the gametes are going to either have one too many or one too few. If the disjunction doesn't happen until meiosis 2, half of your gametes are going to be normal, while the other half are either going to have one too many or one too few, right? So the effects are potentially a lot worse if it happens in meiosis 1 versus if it happens in meiosis. So you have more chances of normal offspring here, less chances of normal offspring here. So all of them have the incorrect number versus only half of them having the correct number. I think this is the last letter we're going to do class today. So when non-disjunction happens, this is an issue. Offspring are going to end up with the wrong chromosome number. So in this case, there was an error when mom made her gamete. She ended up with an extra copy. So when we fertilize that with a normal sperm from dad, the offspring is going to have 2 of each of these chromosomes, but they're going to have 3 of this one. That is called a trisomy. So if you have 3 of one of the chromosome pairs instead of 2, trisomy. Tri means 3. So usually we say this individual is a diploid plus 1. So they're diploid, but they have an extra copy of one of the chromosomes. There are a few examples of these cases that aren't immediately lethal. One of them is probably fairly familiar to you. We'll talk a little bit about it tomorrow. But trisomy 21 is also known as Down syndrome. Okay, so that's one of the more common and probably like the least detrimental health-wise of the trisomies. Okay, there's a couple others that will still not be lethal. But for the most part, if you have 3 of any of your chromosomes, it's going to be fatal right away. Okay, so you're probably not even going to be born if you have a whole extra chromosome minus the 3 cases. On the flip side of that, it's also possible in non-disjunction to pass on a cell that is missing a chromosome. Okay, so in this case, here's the other half of that non-disjunction. So this cell ended up with too many. This cell ended up with not enough. Okay, we're missing one. So when we combine that with the normal sperm, we end up with a diploid cell, but it's missing a partner. So instead of a trisomy, this is called a monosomy, the mono meaning 1. Okay, so, and one of our pairs only have 1 chromosome. So we're diploid minus 1, or diploid except we're missing 1. Okay, this is almost always fatal. The only exception to that is some sex chromosomes. If you're missing if you only have 1 X and no other sex chromosome, you're still able to survive. We'll talk more about that tomorrow. But besides sex chromosomes, if you're missing one of any of the other chromosomes, then that offspring is not going to be viable. They're not even going to make it to birth, or generally they don't need. It's like a spontaneous abortion that's going to happen somewhere along development. So just want to introduce to you those different things. We'll talk about some specific examples of these issues tomorrow. We'll focus a little bit more on sex chromosomes, but I do want to give you some time to finish up your lab today. So go ahead and get that out. Grab your supplies from up here if you want me to take some pictures. This is the last time and cost you'll have to work on it, so make sure you get this done. There is a meiosis lab quiz you need to try to take tonight for homework. Yeah, I didn't set up a group assignment so that everybody can send it in. So if you're done with the lab and haven't taken the quiz yet, make sure you do that.
Oh my gosh.
So I would make sure that you look there to reset the password. But you should be able to sign in then. Okay, you don't need any to do that in the back. You gotta get the stuff on.